heavily, heavily all the samples pretty much, and human polyomavirus, at least 50% of the samples in the middle section have all positive human polyomavirus. And this one is just a Bacteroides showing here again that really this side there was just 2 positive samples, there wasn't really much going on. But when you get to the residential behavior, there is some sort of impairment happening. So conclusion, sewage impairment in the waters, yes. But massive sewage leak, no. I don't think that this is some massive sewage incoming in the system. Markers support temporal and sleeper fringe-based monitoring, true, because look actually on this last slide, you can see that this is the area marker really identified the contamination, but this area that actually was passing by standards is actually good. So otherwise not so bad because of that. And old sewer lines is a universal problem everywhere. I think that's the issue everywhere. Nobody has funds to replace them or fix them or monitor them. I think, and it's just that's the life I think. And to do, we have rainfall data. I looked actually on site 1 and 4 on the rainfall data because of one publication, and I did not see a good correlation between this, and I don't know why. Everyone knows the rainfall and bacteria levels in the stream of control. So microbial communities, we have sequencing right now samples so we could overlay how the microbial communities change around the stream. Origin, I don't know where its origins is sewage. I did not try to identify why is the pipe, or I did not walk the stream. It was really to see how the markers work and how they function depending on the land use, or the rainfall, or how the microbial communities change on the stream. So it wasn't really, I wasn't really trying to fix the problem. More markers is always good. Study Oahu coastline. So this is the second study. So I showed you the rapid results for the Oahu coastline before, but this is the 12 sites that we monitored over 1 year. We sampled every month. So these are the, but I went to Google, no, I didn't Google, I went to the Hawaii water site and I looked what of these species, what of these sites were data available for that time. And so I found that there was data available for these sites, and I found so many samples were collected. And we don't have capability to collect so many samples, but we at least try to go once a week, once a month, and we did always 12. And so this is the data that was calculated based on UH data that's free available on the web. So these are the interval geometric means for these sites over the year. And these are the CPAP frequency count. And this is the state beach action value for enterobacter, but this is used together with was driven by frequency counts. So if you look on the just the enterobacter counts, one of these samples, I think it's 20%, but I think it was one sample that did not compile with the water quality standards, with one standard. But if you look on the both, all the samples should have passed. When we look at our data, so these are our geometric means for these sites. These are our geometric means for CPAP frequency counts. So the beach action value was exceeded in 23% and 50% of samples. I think this is one sample, this is 2 samples, and this is probably 3 samples. And this one, the Kehi Lagoon, we know that we have problem that had also once CPAP frequency standards and the enterovac standards both exceeded. So I think they both actually show the same thing. So what's interesting here is that I need to point out the site number 6, which is Kahala, which is always brown border, and there is always high concentrations of enterovac. And I think there were 3 or 4 samples, 23% of 13 were exceeding the standard. So only a single sample out of 151 samples exceeded both values. So this is this one. That was the 0.6%. If you count for all the samples that we collected. And that's supposed to be 140 samples. 141 samples. It wasn't 151, sorry. 0 samples positive for human bacteroides at site 6. So So this one will be human Bacteroides, and none of the samples contain human Bacteroides. In this one, 31% of samples in KHR wound are positive for human Bacteroides. So we did see human marker in these samples. We did not want human polyomavirus marker, and we think that's because it gets diluted to low detection and concentration. And most important, overall samples indicate good water quality in the majority of each study. So we did not see anything surprising that we don't know. Sample volume. So the current beach monitoring programs evaluate water quality based on 100 milliliter sample volumes, while overhanging evidence shows that microbial concentrations vary vastly in space and time. This is a huge concern as environments such as small water samples not representative of actual water quality. And analysis of larger water samples is needed. So last spring we borrowed an instrument, the PMAX, this ultrafiltration device that allows you to filter 100 liter volume samples. It uses dialysis filters that has a pore size of 150 kilodalton. It is computer controlled. It's engineering very complicated, so I'm very scared of this machine because if something goes wrong, I don't know how to fix it. It has two parts. This is the collection unit and this is the recovery unit that uses the pack rinse the filter because it's then good with filtration system. Everything stays in the filter, it's not getting recycled. And there's a battery and this is the computer on the system. So we took it and we went to the field and we went to the field the water branch, and we went to the field with Street and County Honolulu. We collected all samples from the source water. And we did some sewage spiking experiments. And that's, if you look on all the data, what we saw. Basically, ultrafiltration samples were of course, able to recover, much more likely to recover the different groups of bacteria, like heterotropic total polyforms, E. coli, and Enterococcus, so that's 10% of positive samples. There's a good correlation if you calculate that to 100 milliliter between the quantification of the both methods, ultrafiltration and CRAB. And very importantly, we are able to get, it's always shown here that these are 3 peach sites we studied. We are able to recover viruses and human Bacteroides markers more likely with the ultrafiltration device. And importantly here, we are able to recover PARs actually from the samples. PARs, the RNA PARs is 28 nanometers in diameter, so it's very small PARs. The filter is 150 kilodaltons, so it shouldn't be an issue. The pros, it gives you the representative sample. It's excellent for detection of rare targets, can pick up small viral pathogens. Utilize disposable filters which are reasonably priced. The filter, the browse filter, which is for $200, costs $30. Or actually it costs $22, but we need to modify it to fit the machine. Once it's light, labor and time is intensive. You can probably do 2 to 3 samples per day if you want to recover on the same day. Further concentration steps is needed. So it takes everything and puts it down to 370 mils. And from that, for clean extraction, you probably recover it from the filter. There was a chance with B7 inhibitors, which we saw, especially when we adjusted the pH of the sample to 3.5. And this we need to work on it. And cleanup concern is that it's again, labor and time intensive. It uses bleach and large volume of bleach that you need to recycle in your lab. And how much time I have? I'm going too fast. 20 minutes. Okay, then I can talk about this one. I wasn't supposed to talk about, but this is what I think is important, what I want to really, that's my hobby, kind of. It's the Honolulu wastewater treatment plant. So we have a project. So I think that the public and environmental health concerns are really tightly connected. So we are trying to look on the microbial communities are very sensitive to change. Tools to study microbial communities are matured. So I would say that we can actually now look on the two things together. So we have collected samples from the 4 wastewater regimes 3 times a week. And we have collected samples around the ocean outfall and around the Microbial here. And we have tested all of them for fecal indicator bacteria. We have 200 filters, from which 100 are right now finished sequencing, and we need to send another 100 in. And we have over 1,000 isolates from half to A and naive R, just in case we find something interesting. And here's the data, for example, from the Sand Island, around the Sand Island ocean outfall. So, for example, these are the enterophile levels that we saw in the outfall, and these are the bacterial colonial artery media. And if you look on the coast, then, for example, in this side, sea virus hard surface, we find also high concentration of enterophile, and these bacterial colonial artery, this one. And we also find the finance SS8, which is it's somewhere here. So concentration much higher here. Did they come from here or did they come from here? And I think we can answer these questions by looking at the microbial communities in the sample. So I'm just saying that we're currently working on it, and I think it's important. So we're trying to figure out the special microbial fingerprints that are specific to contamination source. And yeah, I will leave this one. That's all the projects that we're currently doing. We complete Viking beach nourishment project. We complete the slow sand filter study. We look on the this is almost complete, the microbial communities in soil amendments. We look on the type of FIBs and source specific marks in beach, and we're currently working with Department of Health and doing microbial source tracking, actually trying to figure out what is causing the impairment at the Kahalo watershed. And we also have funded right now a small project to study microbial pathogens and microbial communities in the harvested rainwater in American Samoa, and we also work with Susan Yarden and the Big Island and probably Sea Ground. We do some work also there. So that's what's happening. And funding was provided by Hawaii Department of Health, currently for the project with Kahalo watershed. Dr. Richard Cox helped me also, and USEPA funds have been supporting most of the projects that I described, and WRRC is my partner fund. And these have been people who have been helping me throughout the project. I scattered them around, so we know hierarchical because I hate hierarchy. And thank you very much, and mahalo. Any questions?
The PMAX concentrated human polyomavirus.
Yes, we will find it on slides. Yes, it can.
And that's the only concentration mechanism we have.
Right now, yes. There are other methods, but they're costly. And the life, what really right now about Pimax is that it's using these filters that are disposable. I can take my filter and throw it away after I'm done with it. The more expensive needed for cartridges, I think they cost thousands, and you need to clean them and use it again, and how clean you get it. But there is always a question how clean you get the Pimax itself when you're washing it with bleach and sodium thiosulfate and water, so there is always let's clean up and avoid the cross contamination.
But we just say that polyoma is the most human specific.
It's human virus. It can't come from any other source. But the problem is really that it's getting diluted, and the coastal environments are hard to see. At least all samples. We had some of the text here and there, but it's nothing that I would be confident to show you and talk about, because it was levels are low. Even for Manoa beach, from Manoa stream, the levels that showed they were on the borderline between the limit of detection and limit of quantification. So there was really low ones shown. This was, I'm not sure if I can put this in publication. I probably need to show the percentage table. How many percentage positive and negative samples.
So what was the concentration mechanism for Pimax for the polyoma?
For polyomas, the Jody Harwood was the person who really teaching the methods. We use pH to switch to get the virus attached to the. We bring the pH to 3.5 as she recommends. She recommends to use the pH switch to 3.5, and with 3.5 the polyoma virus attaches to the filter. I know that she published this paper with Paris Chapnet, where they did the magnesium chloride, and we found that for polyomavirus, I think the 3.5 the pH adjustment was better. But I know from a PhD project where I adjusted all the samples for magnesium chloride, And it worked 90, I was very successful concentrating polyphars with it. It's like 99.999% polyphars, which is 28 nanometer, stick to the AJ filter with no problem. It was very solid. But did I answer your question? Just we use pH. We use the pH. We do HCl. We had like I don't know 600. We had very small volume of pH like to bring the pH to 3.5 to get them attached to that.
So it is absorbent or is it a vector?
It's absorbed. I think it's charge. It changes the charge of the virus envelope.
You say you use a membrane or you
membrane?
It's a membrane. So
Yes.
water actually passes through the membrane.
Yeah. We also can Pmax.
Yeah.
Pmax does not mean anything. Pmax is just as it is because the pore size is very small. I think the people who actually develop the Pmax, they think that even you can start to capture bigger molecules. I'm not sure what molecules they're talking because I'm against them, that's probably. But if you have something that can capture 150 kilodalton size molecules, you can definitely capture 28 nanometer virus. So there is no adjustment needed. You just take the water, you just throw the tube into the water and just filter from on pH. But how we plan to utilize it is we probably think that we take the big jar and do a crab samples and fill the jar with different from different spots from the pH, so we get a better representation of the whole pH. And we can do it also probably for sand, but we try to test it out. So we do with sand, we mix it up with water, and recover. But it's sand. I'm speculating. I know you're the sand specialist. I was just speculating.
So the virus, do all viruses have similar isoelectric bonds?
I don't know. But for Pmax it doesn't matter because Pmax does not mean any.
Doesn't need
Because just it's done as filters. These are the kidney filters we just bought. They have a hard time actually buying them initially. In some places you can't buy it because it's the medical kidney dialysis filters that we use.
What kind of work are you going to do at Kahalo?
Kahalo? Kahalo, we have ongoing thing. We plan to use the Pmax to recover for pathogens. We are planning to try. There are elevated accounts. There is definitely we have. I don't want to speculate right now on the results, but we are trying to identify the contamination source. There is definitely a problem with human sewage in the system. We know that, and we are working on it. I don't have the data.
Most of these source identification.
Yeah, I can tell you that I'm personally very convinced that there is. But at first, when I first saw the Kahalo map and I saw the wastewater transfer station there, I was so sure that that's the wastewater transfer station. I can definitely tell you that wastewater transfer station is not interested in this anything. It doesn't release to the stream. We sampled along the stream, and we can find human marker above the stream, and the concentrations are high actually.
So you think it's cesspool related?
Kahalo?
Yeah.
I think that it's a combination of different things.
It's interesting that you found that for your coast water samples, there's a lot of inhibition.
It's exactly opposite what we expect. Because usually it's a stream, there is like a humic acid or something, but I don't know what's wrong with it.
So what do you mean by inhibition? You don't get amplification at all, or you just get it?
Also, that slide was actually the true graph from the. I'm trying to go back to that one. That slide that I showed. This is typical graph that we see when you enter water from the coastal samples. This one. So you can get no amplification, or you can get amplification delayed. And it was so dominant in the samples. I don't know why. So if it's more than 3 units, you can't really do it because it's more than 1 log difference in counts. And so most of the samples we go analyze because they're inhibited. We don't use them for the rapid method. Yeah.
What do you mean by inhibited?
It means that it's it means it's this one here. So if we took the samples, we had all. Let's say we have 20 samples. Each sample we put salmon DNA, salmon scalar DNA. They all get same amount of DNA, and for one sample we have water, and we do the DNA, and the water came out somewhere here, and so. When we do the test, they also came up same time because they have same amount of DNA in it. But instead, we're going to detect them. We're going to detect them later. We're going to detect them somewhere here or here. We don't detect them at all. So it means that what happened in PCR in each cycle, the DNA needs to double, and it's supposed to go from 2, from 4 to 8 to 16, but it doesn't. So it's getting delayed on the back, and we will not get the correct concentration. We did it very simple way in samples with salmon scalar as a control. This is one thing that people say is also that it's not linear, that inhibition in this area is, but that's okay. That is, if you put something in here, something so strong, you will never detect inhibition. This area, the PCR is actually more sensitive. So that is also important how much you put it. So it can vary from study to study. But we use the recommendation from the EPA. So the idea was to copy the EPA protocol. Yeah. But the delta CT values that I used to calculate this one are actually delta CTs, not delta CTs. That's a recommendation by EPA.
But you're saying it's from the sand.
The
sand in the sample.
Suspended, something is suspended, I think. Yeah. I don't know, I'm speculating. I have no answer for that. I don't know what I did. Maybe there is something wrong, but Alberto was interested in this one. I went for a couple of samples and we saw it again. Like, just end of last year. We don't know what's happening. And I'm losing Alberto actually short through, unfortunately.
So can you describe how you treat the sample?
Sorry?
How did you treat the samples?
Treat the samples?
Yeah, the sample. How did you concentrate? How did you filter, extract?
100 mils through the PC filter. Nothing else, no pH change, nothing. Just 100 mils to the PC filter as the protocol says. And the tube has already class beads. And then you put the A buffer with the salmon's thermometer unit. And all of them get the same buffer, gives them out. And repeat filter. Then you centrifuge it 3 times to get rid of the solids. Every time you remove some liquid. Another one, spreads the new tube, another one. And at the end you should have the DNA without any loss because everything should stay in the solution. You spin column, which gives you nice clean DNA, there are huge losses to it. So it's MST, when you do spin column and clean DNA, it's getting tricky because polyomaviruses you need to use different type of filter. You can't use this method. And some labs don't use polyomaviruses at all because they can't use this extraction method. Some say it's too much work to change the pH. Yes?
So your method is highly reliant on filtration, but like, do you know any reason like EPA doesn't recommend to like pre-enrich sample?
Pre-enriched samples?
Yeah, to to detect double level.
I don't know if EPA recommends pre-enrichment. I know that the only thing that I know about enrichment from my personal knowledge, I don't know what other people have. I know that the only thing that I know about enrichment is if you take the polyphage and enrich it in the broth for 48 hours, then you take a sample and want to grow up in the broth. There are 4 subgroups of polyphages. 2 of them are usually dominated from animals and 2 are from humans. Put them all 4 together, grow together. One of them, the MS2 phage, growing the fastest because it has the fastest replication. The ratio is getting biased with enrichment if you grow them. That's all that you know.
So you'll show us some sampling from the coastal water,
Yes.
close to the Woody and Sand Island outflow.
Yes.
So I'm not sure, did you say that all the sampling results indicates in compliance
No.
with water quality standard?
No.
There's some violation of the water quality standard?
No.
Oh,
Then I was wrong. Honolulu? No. I never wanted to say that. If I got that then I got it really wrong. No, I'm sorry. No. The project that's ongoing, when we're looking at this one, is they are not in violations actually. The sea prevalence counts are not here, but we should have sea prevalence counts. This one sample from here, which is away from the coast, would be actually the current 130, just based on the enteral results. But everything else was clean. No, I did not. And as I said, I have only analyzed, I'm not saying it here, I only analyzed one sample for markers and all. But actually, all the wastewater treatments, I shouldn't say that, but all the three did actually better. I have the FID data for Hawaii Kai and Kailua and for Honolulu, and I think they clean up the sewage was actually, I think it was higher. That one I showed you was like 97%. Others were close to 100, too low reduction. Initial FID comes through the system. But I'm still doing the purpose of this one is if you get high count somewhere in the middle of here, if you have the information, the fingerprints of the wastewater treatment plants, if you know what's coming out from them, if you know from Hawaii cows come from that, from Hawaii Canal comes from that type of bacteria, from that plant comes from that type of bacteria, you should be able to start to see this, to detect this signal using the community analysis tool. But that might be wrong. I'm just, that's what I'm thinking.
The work still ongoing.
Yes.
At the moment.
This is
The only thing we can say is still water quality generally good.
I put this on here because I just noticed it yesterday that we completed our samples, but I haven't yet got email with our results. But, so, yeah.
Another question I have, earlier you compared Manoa stream and Alawai Canal. Seems you indicated that the marine water is less effective in terms of die-off, bacterial die-off.
Yeah.
You give the reasons. I didn't follow.
The reason was very simple because initially we had freshwater and marine samples. What we did was we filtered it to 0.2 micrometer filter to remove all the bacteria, all the native flora and fauna from the samples. So if we have a wastewater sample and then we look at the wastewater sample that is freshwater type, and we put it into the, if we spike it into the freshwater and if you break it in the marine water, likely the crazes, the fauna that eats bacteria, that eats bacteria, the grazing of bacteria, it's more likely to be it's more likely to succeed in the freshwater than in the marine water because it's from freshwater type source. Because if you take the freshwater animals and throw it in the marine water, that's the simple reason why wind test is different. Another reason that might be is that salts are some sort of buffer. That usually salts are good for the bacteria. If you throw the bacteria into the DI, for example, they just die. But if you have some salts like buffer there in the environment, that might also facilitate the. But I don't know. I'm just saying what might be the reason. I don't know.
So one more question.
On the previous slide, when you were talking about the Honolulu wastewater treatment plant, when you were taking samples, are you taking samples near? You weren't doing any near the outfall way out in the ocean, were you?
No.
These are all along the coast.
Sand Island we did around Ocean Alpha. This is Sand Island.
Oh, the Sand Island.
And we have around Ocean Alpha. These are quarter tanks. We can send out Karatri who helped me to kind of convince the team to get me these samples. And I think it would be really interesting to look at the sediments. I think we could expand our program if we can count on look on sediments, how the microbes in the sediments can change. The way outfall would be really interesting.
Right, but the ones you're taking, are those near shore? are those, where exactly were you taking them and how
So
did you
these
collect
dots
these
are
samples?
these dots are the places where we took the samples. So these like the trajectories. This is the coastline here, and these are the samples for. And these are the samples on the coast. So these are the coastline. These are the standard sites where the city took these samples. And they collected samples for us. And interesting for us, I just want to show you entry levels by city and county and all that are very similar. So we found the same numbers, so that's pretty cool.
Mark,
Yes.
at the same island, the outflow is 80 meters below the water surface.
Okay.
So if you take the water surface sample nearby, you may not catch the impact of the effluent because the effluent may not reach to the surface over there, further away from it.
We have three surface, medium, and bottom, middle, and bottom. So we have 3 levels samples. Each site has 3 samples. So surface, medium, and bottom, but even that might be not good now.
You say you take the deeper water sample?
We did 3 samples at each spot. From the surface, from the middle, and from the bottom. That's water column.
Bottom, how
Bottom.
deep the bottom?
Bottom. But yeah, so that's what I could get. And it's a small project. That's a RIP sponsored project actually. But I have been delaying it. And I thought that I can be really smart, and I developed new primer that was able to sequence a longer fragment, and then I found out that the Illumina platform is not compatible because TM is not right for Illumina. And so I need to build a completely new library, or Roberto helped me with that.
So I think we should stop today. He's our in-house expert, so he's always available at the WRRC if you have any more questions and any queries to follow. So thank you, Marek, and thank you to everyone for